Hello and good afternoon. I am Cody Moon. The date is 4/29/22, and this is my week 3 sensory physiology portfolio assignment. So, the topic I decided to discuss was the anatomy and physiology of the retina. Like I said, I'm Cody Moon, and this is for medical physiology. So first, before we dive into what the retina is, we need to talk about vision. So vision is the process through which light reflected from objects in our environment are translated into a mental image. This can be broken down in 3 steps. First, light enters the eyes and the lens focuses the light on the retina. Secondly, photoreceptors of the retina transduce light energy into electrical signals. Thirdly, neural pathways from the retina to the brain process electrical signals into the image. So, the retina is the innermost layer of the eye. It consists of 2 main layers. The first layer we'll talk about is the retinal pigmented epithelium, or RPE, and the second one will be the neural retina. So like I said, the 2 main layers of the retina. First, the retinal pigmented epithelium, or RPE, is a single layer of epithelial cells on the outermost layer of the retina. This is mainly used for nourishment of the retina, but it also protects as a barrier to avoid unwanted objects into the retina. One cool thing that the RPE does is it protects us from getting blurred vision or messed up vision if there was a situation where light was allowed to move through the cones and rods and miss the cones and rods. The RPE catches those so that those don't affect our vision in day-to-day basis. The second layer is the neural retina. This consists of multiple layers. The 3 main cells present in the retina are the photoreceptor cells, which convert light energy into electrical signals, the bipolar cells, which are the main pathways from the photoreceptors to the ganglion cells, and this is the most direct path for input and output signals. And then the retinal ganglion cells. These process that visual information that we see that begins as light, and they transmit that information to the brain via axons. So this slide here shows you a broken down retina cell. So as you can see, the pathway of light travels into the eye here, and then the light would travel left to right on this diagram. So you can see the ganglion cells, then the bipolar cells. The horizontal cells are kind of a middleman to the photoreceptors. And then you get to the photoreceptors that are the rods and cones. So like I said, the light would travel left to right. And then once that light would travel through to the end where these rods and cones are and the photoreceptors are, then it will travel back right to left and give us that signal output. So what are photoreceptors? Photoreceptors are specialized cells for detecting light. The 2 main photoreceptor cells are cones and rods. There are around 120 million rod cells and about 6 million cone cells in the human retina. So that comes out to be about 6-to-1 ratio for rods and cone cells. So rod cells are mainly located on the outside of the fovea and assist with peripheral vision. The more these rod cells are more sensitive to light than cone cells. These are mainly responsible for low light vision. So anytime that we're trying to see in the dark, that being a dark room or at night, anything like that, that's usually where the rod cells are the most effective in aiding the visual output. So when light levels increase, the rods are unable to effectively change with the light intensity. So what that tells us, as the light intensifies, or we go from nighttime to daytime and the sun comes out, these rods are less effective in the ability to change with that intensity, with the same intensity that the light changes. So then the cone cells are mainly located in the fovea. The central part of the fovea contains no rod cells. So like I said in the last cell, the rod cells are our main peripheral vision cells. However, the cone cells do help a little in the peripherals, but mainly just for the color recognition that we need. Cones are very minimally sensitive to light and mainly responsible for daylight vision. Like I said, the difference in the cones and the rod cells are the rod cells are mainly used for nighttime or dark vision. They help us see blacks, grays, whites, that kind of color, and helps with our peripherals. Where the cone cells are our main providers during the day, and their main function is to give us that color detection. So any of the colors that we see on a day-to-day basis is mainly through the cone cells. A little fun fact about the cone cells are there are 3 smaller cells in the cone cells which help differentiate colors like red, green, and yellow. One thing about the cone cells is people who have color deficiency issues or just have issues separating what colors are, usually it is a cone cell mechanism or reason that they can't differentiate those colors. So here are some sources that I used. Like I said, today was mainly over the retina, physiology, and anatomy. One of the biggest things I have learned through this is there's a lot of small individual cells that take into different abilities to produce the big picture. And that's one thing that I really have stuck on working in the medical field, learning about all these different situations, health concerns with eyes or health concerns with your general body that affect your vision. And it is nice to learn what cells are doing what specific jobs in our vision to kind of see these cells affected with those health issues. And that concludes the anatomy and physiology of the retina.